Hi everyone, this is Reema here, your biology educator from V-Learning. So as you know that V-Learning is an online educational platform where we strive to provide quality education for all. So to get all the benefits from our channel, do subscribe to it, and also don't forget to visit to our website, that is www.v-learning.in, where you will be getting all the PDF notes of this topics and also the animated videos, the pre-recorded videos and the live classes. So let's begin today's session. So in today's class, we are going to start the new chapter, that is your chapter number 10. So in this chapter number 8, you have learned about all the cell organelles and the cell, right? And who has discovered cell and what are the various functions of the cell organ in our body. So we have seen the various cell organelles like the mitochondria, endoplasmic reticulum, Golgi bodies, right? And then the plasma membrane. So we have studied all these things. Now, in this chapter, what you are going to learn is the cell cycle and cell division. So, how the cell divides, okay? The cell, each and every cell, the divides in our body, and for the division, they need to complete one cycle, okay? They need to complete one cycle. That is the sequential events which takes place during the time of cell division. We call it a cell cycle, okay? And you will be learning about the cell division, okay? How the cell divides. So, you know that how a baby is being born, right? It is by the fusion of gametes, isn't it? From the male and the female body, right? So, the male and the female gametes, they fuse together, they form a zygote, and then you know that the zygote, it leads to the formation of embryo, right? And further the embryo divides, that is the cell divides. So, each and every organism each and every organism are being born by the process of cell division, okay? So you know that the omnicellular, right? So what it means the each and every living organism are being formed from a pre-existing cells, okay? That is, they are being formed by the process of cell division. So in this chapter, you are going to learn about the cell division process. That is, the cell divides by the process of mitosis and meiosis, okay? The cell divides by the process of mitosis and meiosis. So you will be learning about the various phases. What are the phases in the mitosis and the meiosis? And also we'll be learning about the process of cell cycle and what are the process how the cell divides. So let's begin today's class, and this is your lecture number 1. So in today's class, you will be learning about mitosis. Okay, what are the various steps in the process of mitosis, and also you will be learning about cell cycle, right? So let's start the class. So see, what is the cell cycle? Now, you know that each and every organism, we are made up of cells, right? Our body is made up of millions and millions of cells, isn't it? So, the cells, they need to divide, okay? During the time of replication, the cell divides, okay? So, how the cell divides? By the division of chromosome, okay? So, the chromosome, it divides. So, if this is a cell, okay? If this is a cell, the cell will undergo division, and what it will form? It will form two daughter cells, right? It will form two daughter cells, isn't it? Now again, from this two daughter cells, again the division will occur, and it leads to the formation of four daughter cells, isn't it? So that means again the cell will divide, right? So how many cells are there now? You can see that 1, 2, 3, 4, 5, 6, isn't it? So this is how the cell divides. So this cell will lead to the division, and it will form again what? Daughter cells, isn't it? So it is forming the daughter cells. So this process, okay, the sequential process, how the cell divides, okay. So in case of human beings, we have what? 23 pairs of chromosome, right? So all the chromosome in our body is diploid except the chromosomes, that is the sex chromosomes, they are haploid, isn't it? So the total number of chromosome is 46. Okay, the total number of chromosome is 46. So when cell divides, what happens in that case? The splitting of the chromosome takes place. Okay, the splitting of chromosome takes place. So the sequence of events by which cell duplicates, as you can see, how the cell is being multiplying or it duplicates, isn't it? That is called what? Cell cycle. The sequence of events by which the cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells. Okay, two daughter cells is termed as cell cycle. So what I have explained here, how the cell duplicates itself. Okay, so what is being duplicated in case of cell division? In case of cell division, the chromosomes. Okay, the chromosomes, it divides. Okay, it duplicates. Okay, it multiplies. All right. So in case of cell, the chromosome, it multiplies, it duplicates. Okay, so what happens in that case? The daughter cells are being formed. All right. So see, the DNA synthesis occurs during only one specific stage in the cell cycle. So the DNA synthesis, it occurs only in one specific stage in the cell cycle. Okay. So in the cell cycle, we are going to learn now that there will be two phases basically. One is interphase, another is mitosis. Okay. So in the interphase, you will see that there are again three phases. So in that three phase, you will see G1, S and G2 phase. So basically, the DNA synthesis takes place in the S phase. Okay. So the DNA synthesis takes place in the S phase. So in the S phase, the DNA synthesis takes place, and further it undergoes cell division. We will learn it in the next topic. So let's see. Now, what is the cell division? Why it is very important? Okay. So this cell division is very important. Why? Because it is necessary for the formation of a new living organism. Okay. So it is responsible for cell division, for DNA replication, right? And also for cell growth. So it is an important process in all living organisms because during the division of cells, the DNA replicates. Okay, the DNA replicates. So number one is replication of DNA, then cell growth also takes place, and all this process, that is the cell division, then DNA replication and the cell growth, can have to take place in a coordinate way in order to ensure what correct division and formation of progeny cells. So as I said, that continuation of the generation means that the formation of embryo takes place by the fusion of male and female gametes, isn't it? So from that, what happens? That will lead to the process of reproduction, isn't it? So it leads to the formation of progeny cells, right? So when cell division takes place, let's say this that is the male and the female, right? So the fusion of gamete is taking place. Okay. So fusion of gamete takes place. So fusion of gamete takes place, that is haploid. Here the gametes are haploid gametes, right? So what happens? N number of chromosomes from here, N number of chromosomes from here, and it will lead to the formation of diploid, isn't it? So this is diploid. Now what happens? This will form the zygote, that is fusion of male and female gametes, which leads to the formation of zygote. We call this process as fertilization, right? We call this process as fertilization. So this is the process how the fertilization takes place. Now from this zygote, what happens? This is the progeny cell. Okay. Now this needs to divide, undergoes again division. So here what happens? Here now it is two cells, isn't it? Now it is two cells, that is the sperm and the ova, isn't it? So here it is two cells now. Now again the cell further divides. From the two cells, it will be forming what? It will be forming the fourth cell, isn't it? It will be forming 4 cell. From two cell, it will be forming 4 cell, right? Now again from the 4 cell, it will be forming what? It will be forming 8 cell. Clear? This is how the daughter cells are being forming. Okay, 1, 2, 3, 4, 5, 6, 7, 8. One more. Okay. Yes. Now see, so here 8 cells are being formed, right? 8 cells, okay. So it is leading to the formation of what? Embryo. So embryo formation. How it is being formed? The process of cell division, right? The cell is dividing, okay. The cell is dividing. Now, this cell division is very, very much important, okay, because it leads to the formation of what? Progeny cells. So these are all what? These are all the progeny cells, okay. From that, fetus is being developing, right? So a baby is being born by the process of cell division, okay. A new organism is being formed from the pre-existing cells only. So what are the pre-existing cells here? The parent cell, right? So these are the pre-existing cells, and from here the progeny cells are being developing, isn't it? So this is a very important process. The cell division is a very important process for the formation of the progeny cells. Clear? Okay. So yes, and it contains the intact genome. Okay. Intact genome. So this child, okay, this progeny will be having the genes of the parents only, that is the father and the mother, okay. So he or she, that is the progeny cell, will be having the genes of the parents only, okay, because it is what fusion of gamete is being taking place here, right? Now, what are the various functions? Okay, what are the various functions of the cell division? Why it is necessary? Because during this process, what happens? The DNA replication takes place. Okay, that is the replication of DNA takes place, right? So, what happens? The chromosome. Okay, the chromosome. It leads to the formation of another pair of chromosome. Okay, the chromosome replicates here, and the cell growth. Okay, the cell growth takes place. So that's why it is an important process, the cell division. Yes. Now let's learn the phases of cell cycle. So as you can see in this picture, okay, this is from your NCERT. Here you can see G1, right? Then S, G2, and this is M phase. Okay, this is M phase, mitosis phase, and here this is G0 phase. Okay. Now what is this? Okay. So the cell, for the cell to divide, it undergoes basically two phases. Okay. So cell division or the cell cycle. So what are the various stages of the cell cycle? The cell cycle takes place by following the two stages. One is your interphase. One is interphase and another is M-phase, that is mitosis phase. Okay. All right. Now, in the interphase phase, okay, the cell prepares itself. Okay, the cell is not dividing, the cell is preparing itself for division. So, let me tell you, a human cell, it requires 24 hours to complete the cycle and divide. Okay, how many hours it requires? Human cell requires 24 hours. Okay, 24 hours for the process of cell division. In order to complete all this cycle, it completes all this process, okay, and then it divides. So for completion of all this process in case of human cell, it requires 24 hours. So the process is different in case of different living organisms, okay. So different living organisms, the process of cell cycle and the cell division is different, okay. So in case of human being, it is 24 hours. Now here in interphase, in the interphase itself, it takes 23 hours. So in the interphase only it takes 23 hours, and in the mitosis phase it takes only 1 hour, because here the cell is already being prepared. So this interphase is the preparatory phase. Okay, what is this interphase? This interphase is the preparatory phase. All right. So here the cell prepares itself for division. It is the preparatory phase. And here in the mitosis, what happens? The actual cell division takes place. So in the mitosis, what happens? The actual cell division is taking place. All right. So the actual cell division is taking place in the mitosis. Okay. Now, in the interphase, there are again three phases. Interphase is divided into again three phases. So, what are the three phases? Yes, G1. Now, what do you mean by this G1? G1 means gap. Okay, gap phase. All right, gap phase. Then then S phase. Yes, S phase. Then S phase, and then what? Then G2 phase. Okay, so these are the three phases of the interphase. All right. Now here in the mitosis again it follows four phases. Okay, the cell divides by following the four phases. Now what are the four phases? The four phases is like prophase, metaphase, anaphase, and telophase. You can see here, right? So the four phases are the prophase. So prophase, metaphase, anaphase, and telophase. Okay, telophase. So these are the four phases of the mitosis. Okay. So see here again you can see that G0 phase. Okay, what is the G0 phase? Now there are some cells. Okay, there are some cells which do not divide. Okay, after reaching to the G1 stage, after reaching to the G1 stage, there are some cells they don't want to divide. Okay, so they just exit. They exit from the G1 phase. Okay, they exit from the G1 phase. Okay, so that phase is known as the G0 phase. Okay, this phase is also known as the quiescent phase. Okay, here the cell does not divide. Okay, in the G0 phase, the cell does not divide. There are some cells like the heart cells and the nerve cells, the muscle cells, okay? So they don't want to divide. They stop dividing after a certain period of time. After reaching to the G1 stage, they do not undergo this S phase, okay? So this is the phase where actual replication of DNA takes place. So this is the phase where the replication of DNA takes place, okay? So what happens in the S phase? Here the replication of DNA takes place. Okay, that is the DNA replication. Actual division takes place in the S phase. Okay, and in the G1 and the G2 phase, the protein synthesis takes place. What happens here? The protein synthesis takes place. Okay, the protein synthesis. The protein synthesis takes place here. Okay, here also the protein synthesis takes place. Now, in the M phase, okay, in the M phase, further the cell undergoes division, okay, and it leads to what? It leads to the formation of the two cells by the process of cytokinesis. That is the division of the cytoplasm takes place, okay. So, after the cell completes this stage, then what happens after the cell completes these four phases? Then cytokinesis takes place. Now, what is this cytokinesis? So, this cytokinesis is a process by which the cell, that is the cytoplasm of the cell divides. Okay. So, what happens if this is a cell which is divided? Okay. So, the cytoplasm, okay, they will form two daughter cells here, right? That is the cytoplasm divides. Okay, so that is known as cytokinesis. We will learn it in the next topic. So this is the phase. Okay, this is how the cell cycle takes place, and it takes what? It takes how many hours? It takes 24 hours. Now, yes. So this already I have explained that how the what are the various phases of the cell cycle, right? So next, yes, let's see that a eukaryotic cell is illustrated by human cell in culture. So the scientists have done the culture of human cell, and from that they found that the cell divides. Okay, the cell divides. So how the cell divides? In case of human cell, it takes 24 hours. Okay, the cell division takes place 24 hours. That is, the phases of cell cycle is 24 hours. However, the duration of cell cycle, it will vary. Okay, that is, it is different in case of living organism. Okay, so the cell cycle is different in case of other living organism. It varies from one living organism to another. So in case of yeast, okay, in case of yeast, which is a fungi, so what is the duration for the cell? The cell cycle, it requires what? It requires about 90 minutes. Okay, in 90 minutes only, the cell completes its cycle. Okay, so in case of yeast, the cell completes the cycle only in 90 minutes. All right. But in case of human being, how many hours? It requires 24 hours. Clear? Yes. The cell cycle is divided into two basic phases. That is, we have already learned what are the two basic phases: interphase and the anaphase, right? And anaphase. So here you have seen that cytokinesis, right? Now, after completion of all the phases, that is anaphase, the cell needs to go for cytokinesis, right? Now, what is this cytokinesis? Okay, so what are the various phases? Now, you have seen that in the M phase, what are the various phases in the M phase? That is prophase, right? Then metaphase, isn't it? Then anaphase and telophase, right? Anaphase and telophase. So, for example, if this is a cell, okay, this is S phase. This is S phase where the DNA gets replicated. Okay, as you know that in the S phase, the DNA replication takes place. So the DNA replication takes place where? In the S phase. Now, further what happens? It will undergo the mitosis phase. Okay, that is, first phase in the mitosis is the prophase, right? So in the prophase, the cell will be here lying, right? Now it will further undergo which phase? It needs to undergo second phase, that is metaphase, isn't it? So in the metaphase, the cell will lie in the center. Okay, the cell will lie in the center. So in the metaphase plate they will lie, or it is also called as equatorial plate. Okay. So further in the anaphase, what happens? The cell, that is the chromosomes, will move towards the opposite poles. Okay, the chromosome moves towards the opposite poles. As you can see here, the chromosome moves towards the opposite poles in the anaphase stage, right? Then in the telophase stage, what happens? The cell further, the cell further divides. Okay, so it will lead to the formation of what? It will lead to the formation of two cells. Okay, it will lead to the formation of two cells by the process of cytokinesis. Okay, cytokinesis. So cytokinesis is a process where the cell divides. Okay, and the two daughter cells are being formed, as you can see here. So again, the two daughter cells are being formed here, right? So this is how the cell division takes place. Okay, this is how the cell division takes place. It starts from the S phase where the DNA replication takes place, and it followed by the M phase. Okay, followed by the M phase, and this is cytokinesis is the process how the cell divides and the daughter cells. So these are what? These are the daughter cells, right? These are the daughter cells, isn't it? So this is how the cell needs to undergo this division in order to complete the phases of cell cycle. Okay. So again, further this daughter cells, okay, this daughter cells again it will go under the same process. Okay, it will under the same process, same thing. Okay, and then it will further duplicate its number, right? So this is how the phase of cell cycle takes place. So now Yes, interphase. So as you know that there are two phases in the cell cycle. So what are the two phases? One is the interphase and another is the aphase, right? So the interphase, it is called as a resting phase, okay? It is also called as the resting phase. Now why it is called resting phase? Because it is the phase where the cell is preparing itself for division, okay? Here the cell doesn't divide, right? So here the cell prepares itself. So what are the various three phases in the interphase we have learned? What are the three phases of the interphase? It is also called as what? It is also called as the resting phase, right? It is also called as the resting phase. Now here in the resting phase, what happens? The cell prepares itself. So it is also known as the preparatory phase. Now, why it is known as preparatory phase? Because the cell prepares itself for cell division, right? So, what are the various three phases? Okay, what are the various three phases of the interphase? The three phases of the interphase are. So, what are the various three phases? The three phases are the G1 phase, S phase, and the G2 phase, right? So these are the various three phases on the interphase. So just remember this point, okay? So yes. So see, this is for preparing. Okay, the cell just prepare itself for division. So for growth and DNA replication in an orderly manner, right? So the cell prepares itself for the growth and DNA replication. Here the cell is not dividing, all right? It is just preparing itself. Now the interphase is divided into, as you can see, we have already learned G1S and G2 phase. Okay, so this is all about interphase. Now let's move into the G1 phase. As we know that G1 is what? G1 means gap 1 phase, right? So in the gap 1 phase, what happens? The protein synthesis takes place. Okay, the protein synthesis takes place and it is providing nutrition to the cell for the growth and division. So it is what? It is between mitosis and initiation of DNA replication. So as you can see that this G1 phase where it is, yes, the G1 phase, you can see the G1 phase, right? So it is between the mitosis, that is the M phase and the S phase, isn't it? So again, the G2 phase is also between the M phase and the S phase, right? So it is in between, right? It is in between the S phase and the mitosis phase, right? Yes. During G1 phase, the cell is metabolically active. So this is important. The cell is metabolically active. It is very, very active here, okay? And continuously grows, okay? But does not replicate its DNA. So in the G1 phase, DNA replication is taking place or not? No, right? So in the GNA phase, DNA, that is no DNA replication. No DNA replication. What happens in the G1 phase? The cell grows. Okay, the cell growth or the cell grows in number. Then what happens? The protein synthesis takes place here. The protein synthesis takes place here in the G1 phase. So yes, so where it is in between the mitosis, that is Anaphase, and the S phase. Okay. So in the S phase, let us now learn about the S phase. Okay. So what happens in the S phase? In the S phase, the DNA replication takes place. So this is the phase where the cell, where the DNA replicates. Okay. So this is the phase where the DNA replicates. That is DNA replication. The DNA replication takes place in the S phase. Okay. So now, yes. So it marks the period during which DNA synthesis of replication takes place, and during this time, the amount of DNA per cell doubles. So here what happens? The amount, okay, amount of DNA gets doubled. Okay, the amount of DNA increases. So remember, in case of the cell cycle or the cell division, the chromosome number remains same. What happens? the chromosome number remains same, but what happens, only the DNA doubles its number. Okay, the DNA that is the amount of DNA doubles, but the chromosome number is same. Okay, all right. Yes. So here we can see that the amount of DNA per cell doubles. Okay, the DNA increases. Okay. So what happens again? The DNA synthesis takes place. So these are the 2 points. One is DNA synthesis, another is amount of DNA doubles. Yes. Now, in case of animal cells, during that phase, the DNA replication begins in the nucleus. Okay, we will see. Okay, so the nucleus is the place where the DNA replicates and the centriole duplicates in the cytoplasm. Okay, now what is this centriole? Now, this centriole is present. Centriole is present only in case of animals. Okay, in case of animals only the centrioles are present. It is absent in case of plants. So inside the cell, okay, you will see some like this structure. Okay, like this structure. These are known as centrioles. Now what the centrioles do is, the centrioles, they help in the division of the chromosome. So what it does, it helps in the division of cell, you can say, by division of cell by dividing the chromosomes, okay, or by splitting the chromosome, we can write, okay, or by splitting the chromosome. All right. So, these centrioles which are present in animals, they help in the process of cell division. Okay. So, during the G2 phase, what happens again? So, during the G2 phase, here also the protein synthesis takes place. Okay. Here also what happens? The protein synthesis takes place. The protein synthesis takes place, and it is also the preparatory phase, and it is present in between the mitosis and the Anaphase. Okay, so preparation for the mitosis while cell growth continues. Okay, so during the G2 phase, what happens? The cell growth also takes place. So it is also responsible for what? It is also responsible for the growth of the cell. Clear? So this is all about the G1S and the G2 phase, which we have learned under the interphase. Okay. Now let's see what is the G0 phase. I have explained that there are some cells. Okay. There are some cells after reaching to the yes, it is like this, right? So here it is G1 phase, S, then G2 phase, then this is M phase. Now there are some cells after reaching to the G1 phase. Okay, after reaching to the G1 phase, they don't want to divide. Okay, so they just exit. All right. So they just exit and they reach to a phase that is known as the G0 phase. All right. So G0 phase is also known as what? It is also known as the quiescent phase. Okay, because here the cell stops dividing. All right. So there are some cells in the adult animals do not appear to exhibit division. So example is our heart cells. Okay, they do not divide after a certain period of time. So and many other cells divide occasionally. Okay, there are some cells like nerve cells. Okay, so they divide only occasionally. Okay, so as needed to replace cells, that have been lost because of injury or cell death. So this G0 phase also responsible for So there are some cells, as you know, that which will be injured, okay? So they go to the G0 phase, okay? So these cells do not divide. So these are the cells which do not divide further and exit the G1 phase. So some of the cells, for example, the injured cells, okay? So they do not divide and they exit the G1 phase. So they enter an inactive stage called the quiescent stage. So remember this, G0 phase is also known as the quiescent stage. Okay, it is also known as the quiescent stage. So cells in this stage remain what? Metabolically active. They are metabolically active, but they will not divide. All right. So they are metabolically active. The cells are metabolically active, but they will not divide. So they will no longer proliferate unless called on to do so, depending on the requirement of the organism. So these cells will just remain, okay, inactive, but they will not divide further unless it is required, okay, unless it is required by the organism. So this is all about the G0 phase. So next, as we are talking about the DNA replicates, as I have said that in the S phase, the DNA replicates, okay, and we are talking about chromosome. Now, you might be thinking that where the chromosome is present. So, let me tell you that yes, if this is the DNA, now how this DNA is being, from the DNA, the chromosome are being formed. So first let us see that where the DNA is being present, okay. Inside the cell, if this is a cell, inside the cell what is present? Inside the cell, the nucleus is present, right? Inside the cell, the nucleus is being present. So inside the nucleus, the DNA is present. Clear? Inside the nucleus, the DNA is present. Now in this DNA, the chromosomes are present. In this DNA, what is present? The chromosomes are present. Now in this chromosome, what is present? In this chromosome, the genes are present. Clear? In this chromosome, the genes are present, okay, which are the hereditary material. So see how these chromosomes are being formed from the DNA. So see, this is the DNA. Now, as in the class, in your class 12, you will be learning that the chromosomes are being formed when the DNA, it tangles over the histone proteins, okay. It tangles over the histone protein during the process of it tangles over the histone during the process of DNA replication. So during that time, the small beads like structure will be formed, okay. These are known as what? These are known as chromatin. So small bead like structure will be formed. These are known as what? These are known as chromatin. Now further, this chromatin will be entangled, okay, and it will lead to the formation of a chromosome, okay. This is how the chromosome look like, okay. So chromosome is formed from where? Chromosome is formed from the bead like structure, that is the chromatin, okay. Chromosome is formed from the bead like structure, that is the chromatin. So this is the exact structure of the chromosome, okay. This is the exact structure of the chromosome. Now what is this? This is known as what? This is known as chromatid, okay. This is known as chromatid. This is also chromatid. Now what is this then? This is known as centromere, okay. This is known as centromere, okay. Now when the chromosome replicates, during the process of replication, we will see another chromosome here, right? We will see another chromosome, that is the replication has taken place. So these chromosomes are having the equal number of DNA. So they are also known as these two chromatids. As you know, this is chromatid, right? These two chromatid, they are having the equal number of DNA. They have the equal number of DNA, and they are known as what? They are known as the sister chromatids. They are known as sister chromatids. Clear? So this is how the chromosome looks like. This is the structure of the chromosome, and the chromosome is formed from the DNA, where the DNA is present inside the nucleus. So this is how the chromosome is being formed from the chromatin. So the DNA, it tangles over the histone protein, so it forms the bead-like structure which is known as chromatin. The chromatin further tangles over, okay, and it leads to the formation of what? It leads to the formation of the chromosome. Now, this is not the structure of the chromosome. We cannot see this type of structure of chromosome in our body. The chromosome will be like this, okay? When we see under the microscope, we will see this type of structure. But this is shown as like how the chromosome looks like, okay? Because it has got various parts like centromere. Centromere is the place where the chromosome divides, okay? It helps the chromosome to divide with the help of kinetochores, okay? So this is known as chromatid, and this is also chromatid. So when the chromosome replicates, okay, when the chromosome replicates, as you can see, a pair of chromatid are being formed here, right? So these chromatids are known as sister chromatids, and they contain the equal amount of DNA. Clear? So this is about chromosome. Now, how many chromosomes are there in our body? We have 23 pairs of chromosome. We have 23 pairs of chromosome. That means total we have 46 chromosome in our body. So out of this 46 chromosome, 22 are 22 chromosomes are what? 22 chromosomes are autosome, right? 22 chromosome are autosome, and the other chromosome, that is the one, is your sex chromosome, XX or XY in case of the male. So this is what? This is the sex chromosome. Chromosomes are further divided into two types, right? So what are the two types? One is the autosome and another one is the sex chromosome. So in case of male, what will be the chromosome number? That is 22 plus XY, right? 22 plus XY. XY is what? XY is the sex chromosome. And in case of female, what will be the number of chromosome? That will be 22 plus XX, right? 22 plus XX. Clear? So this is the number of chromosome in case of the female, okay? In case of female, this is the number of chromosome, and in case of male, this is the number of chromosome. So this is all about chromosome we have learned. Now let us see how the chromosome, it helps, that is DNA replication takes place in the M phase. So now we are moving into the M phase where the cell further divides, okay, and the cytokinesis takes place. So see, this is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. So this is the most dramatic period where the cell further reorganizes themselves, okay? Now what does it mean? The cell need to arrange the cell organelles for the process of cell division. So since the number of chromosomes in the parent and the progeny cell is the same, so here the number of chromosomes in the parent cell and the progeny cell, okay, and the progeny cell is what? Is same. Clear? Is same. So it is known as what? It is also called as equational division. So why it is called as equational division? This is called as equational division because the parent cell and the progeny cell number is same. Clear? So yes, now let us see what are the various phases of the M phase, that is prophase, metaphase, anaphase and telophase, right? So these are the various phases of the M phase. So of the mitosis. Now let us learn what happens in the prophase, how the cell divides. So in case of prophase, yes, in case of prophase, there are two prophase, the two prophase we will see. One is the early prophase, okay, one is early prophase, and another is late prophase. Okay, another is late prophase. Now what happens in case of the early prophase? Now in case of the early prophase, if we see, this is the cell, okay, this is the cell, and inside the cell you will see this is the nucleus, right? So what happens here, the beginning of the chromosome, that is the condensation of the chromosome begins. So in this early phase, the condensation, okay, the condensation of chromosome begins, okay, and here the chromosome, as you can see, they will look like thin thread-like structures, okay, they will look like thin thread-like structure. Here the chromosome you can see as thin thread-like structure. Now in the late prophase, what happens? In the late prophase, the chromosome condensation takes place, okay. So in the late phase, this is a cell, and here the nuclear membrane starts to disintegrate, okay. Here the nuclear membrane, this is the nuclear membrane, this is nuclear membrane. So what happens, the nuclear membrane starts breaking, okay, the nuclear membrane starts breaking. So the nuclear membrane starts disintegrate, and you can see the chromosome as like this, okay, that is the replication. After the replication, so we see the chromosome as like this, the chromosome condensation has taken place. So you can write that chromosome condensation. So here what happens, chromosome condensation takes place, okay, chromosome condensation takes place. Next, what you can write here, that nuclear membrane disappears. So nuclear membrane disappears. So as the nuclear membrane starts disappearing, all the cell organelles like the mitochondria, Golgi bodies, okay, they will also start disappearing, okay. So as the nuclear membrane is disappearing, they will move out of the nucleus, okay, and they will also start disappearing. So here you will find the centriole, okay. So here you will find the centriole, and they will be having the spindle-like structure. They will further help the chromosome in the arrangement, okay. So here you will find the spindle-like structures, okay. That is called as ester. So here the ester formation also takes place, okay. So this, what happens, this centrioles, okay, this centrioles, it will help in the further arrangement of the chromosomes in the metaphase stage, sorry. So this centrioles, what it does, they will further help the chromosome to arrange in the metaphase stage. So now let us move into the metaphase. Now what happens in the metaphase, as you have seen that in the prophase, the condensation of chromosome has taken place, then the nuclear membrane starts disappearing, and as the nucleus membrane starts disappearing, all the organelles like the mitochondria, Golgi bodies, endoplasmic reticulum, they will move out of the cell. They will also move out of the nucleus. They will move out at the corner, and they will also start disappearing, and the ester formation takes place. So these are the 4 points under the prophase. Now under the metaphase, what happens? So this is a cell, okay, this is a cell. Now here the chromosome lies in the equatorial plate, okay. So the chromosome lies in the equatorial plate. So here the chromosome can be seen, okay. In the metaphase stage, the chromosome can be seen under the microscope properly, because the condensation of chromosome has taken place properly here, okay. So here this is the phase where the chromosome can be visible easily. Now here again you will see the centrioles, okay, and this and what is this? This is the these are the microtubules, okay, and then these are the spindle fibers, okay, these are the spindle fibers. So they are what? They are helping the chromosome to arrange in the metaphase plate. So these are what? These are the microtubules, and this is the centriole. This is the centriole. This is what? These fibers are protein fibers, which are known as the spindle fibers, okay. These fibers are known as spindle fibers. So here you can see the formation of spindle fibers, and with the help of kinetochore, okay, here you will see what? You will also see kinetochore, okay. So here you will also see kinetochore, microtubules. So this you can see the structure, thread-like structure, these are known as the microtubules, okay. These are also known as this is known as microtubule. Here you can see the formation of microtubule, which helps the cell to properly arrange in the equatorial plate. So as you can see, the cells are properly arranged, right, in the equatorial plate. So this plate is also known as the metaphase plate, okay. This is also known as the metaphase plate. Clear? So this is the phase where the chromosomes are clearly visible. Here the chromosomes are what? Here the chromosomes are clearly visible, okay. Here the chromosomes are clearly visible under microscope, okay. Clearly visible under the microscope. Clear? So this is all about the metaphase stage, okay, how the cell arrange. Now let us learn about the anaphase stage, okay. Now after the cell is arranged in the metaphase plate, now what happens? The spindle fibers, what you have seen here, the spindle fibers, it begins to pull this chromosomes, okay. It begins to pull these chromosomes towards the opposite pole, okay. So let us see what happens here. As you can see, this is how the metaphase plate, right, metaphase stage, sorry, this is how the metaphase stage. Now what happens in yes, this is how the metaphase, yes, 2 times it is, okay, fine. So this is how the metaphase here, right, here the chromosomes are here, right. So yes, now in the anaphase, what happens? The cell moves towards the opposite poles, okay. The chromosome moves towards the opposite poles, okay. Here the chromosome, it moves towards the opposite pole, and here you will find what? You will find the centrioles, okay. So here you will find what? You will find the centrioles and the microtubules and the spindle fibers, okay. And the spindle fibers, it pulls the chromosome. Now you will tell that these are chromatids. These are no longer chromatids. These are now called as chromosome. These are called as one chromosome. So this chromosome, it moves towards the opposite poles, okay, opposite poles. So what happens here? Basically, this is the centromere, okay. This is a chromosome, isn't it? So here, this is the centromere. So this is the centromere, right? So as you can see, the centromere, this centromere is splitting, isn't it? So the splitting of centromere takes place. So what happens here in this phase? The centromere splits, okay? The centromere splits here, all right? So that is the splitting of centromere takes place. So this is how the arrangement of chromosome takes place in anaphase. Now after this anaphase, what happens? After the anaphase, the next phase is the telophase. Now what happens in the telophase? The cell will be further dividing, and the nuclear membrane starts begin, okay. The nuclear membrane is being formed again, and also we can see the various cell organelles, okay. The cell organelles also we can see, like mitochondria, Golgi bodies, they also start forming, okay. Now here we will see the chromosomes again, okay. The condensation, that is the decondensation of chromosome takes place, okay. The chromosome starts decondense. So this is how. In the telophase, we will see the two cell. Okay, the cell further it will divide by the process of cytokinesis. Okay, cytokinesis. So after the telophase, what happens? Here the two daughter cells are being formed, right? So here in the mitosis, how many daughter cells are being formed in the telophase stage? The two daughter cells are being formed. Clear? So this is all about the various phases of the mitosis. So we have seen the prophase, metaphase, anaphase and telophase. Clear? So now let us see the picture from your NCERT. See, this is the early phase. As you can see, the chromosomes are here. The long thread-like structures, right? That is the condensation of chromosome has begun. Now here in the late prophase, as you can see, the chromosome has already condensed, and further, the chromosomes are arranged with the help of the spindle fibers and the microtubules. So the centrioles, it helps in the arrangement of the chromosome in the metaphase plate. So see here the chromosomes lie in the equatorial plate. So here the spindle fibers and the microtubules, they have pulled the chromosome, they have arranged the chromosome, and now here the chromosome lies in a metaphase stage. So further what happens, the spindle fibers, it pulls the chromosomes towards the opposite pole. So as you can see, towards the opposite pole, the chromosome has been pulled by the spindle fibers and the kinetochore fibers. So it moves towards the opposite pole, and further the two cells are being formed by the process of cytokinesis during the, that is after the telophase stage. So this is how the cell cycle takes place. And again, after the mitosis stage, what happens? The interphase begins. Means after the M phase, we will see here. So as you see the diagram of the cell cycle, you see here G1, right? Then S phase, isn't it? Then G2 phase, and then M phase. So here after the replication has taken place, so after the M phase, what is this phase? This phase is again the interphase, right? Again the interphase begins. That means again the cell starts preparing itself. So again the interphase, that is cell will undergo the G1 as G2, and then further again it will divide by following the M phase. So this is how the cell cycle takes place. So this is how the cell continues dividing. So yes, now let us learn the M phase, what is given in your textbook. So prophase of the M phase. So prophase, which is the first stage. Okay, so this is the first stage, follows by S and the G2 phase of the interphase. Now in the S and G2 phase, the new DNA molecules are formed and not distinct but intervened. Okay, as you have seen that in the prophase stage, that is in the S and the G2 phase, the chromosomes, that is the DNA molecules are being formed, but we cannot identify themselves properly. Okay, so they are not properly distinct. Okay, so prophase is marked by the initiation of condensation of chromosomal material. So number one point, what we have learned, that the condensation of chromosome takes place, isn't it? So this is the first point, that is the condensation of chromosome takes place in the prophase, and the chromosomal material becomes untangled during the process of chromatin condensation. So the chromosome starts untangled during the process of chromatin condensation, and the centriole, which had undergone duplication during S phase of interphase, now begins to move towards the opposite poles of the cell. So here you have seen the centrioles. As you can see, these are the centrioles. Okay, these are the centrioles. So this centriole, now you can see that after the chromosome condensation, it begins to move towards the opposite pole, right? So it starts preparing for the metaphase stage. So now the completion of prophase, after the completion of prophase, we can mark down the following events taken place. So what are the following events taken place? The chromosomal material condensed, we have already seen. Then the chromosomes are seen to be composed of two chromatids attached together at the centromere. So you have seen that this is the chromosome, right? So these are what? These are the chromatids, isn't it? This is the centromere, this is the centromere, and this is the chromatid, right? So they are being attached with the help of what? With the help of the centromere. So initiation of the assembly of mitotic spindle, the microtubules, the proteinaceous component of the cell cytoplasm help in the process of this. So what are the various things which helps in the process? That is the microtubules and the mitotic spindle. Yes. Now what happens in the metaphase stage? Let us read. At this stage, the metaphase chromosome is made up of two sister chromatids. Okay. Now, what do you mean by the sister chromatids? So, if this is a chromosome, okay. Now, these are what? These are the chromatids, right? So, you can see there are two sister chromatids, isn't it? So, sister chromatids. So, these are the chromatids, right? So as there are two chromatids, so we called it as sister chromatids, so you call it as two sister chromatids in the metaphase stage, so which are held together by the centromere. You know that this one is centromere, isn't it? So it is held together by centromere. So hence the metaphase is characterized by all the chromosomes coming to lie at the equator. So we have already seen that here the chromosome lies in the equator and which is connected by the kinetochore to the spindle fibers. So these are the two fibers. So what are the two fibers? One is your kinetochore, one is kinetochore, and one is kinetochore fiber, and the another is what? Another is the spindle fiber. So these are the two fibers which helps in the connection of the chromosomes. Now, next what happens from one pole and its sister chromatid connected by its kinetochore to the spindle poles from the opposite pole. So as you can see in this diagram, okay. So see in this diagram. So these are what? These fibers, okay. These fibers are helping the chromosomes to connect. So one is at one pole, isn't it? Another is at another pole. So these are the kinetochore fibers and this is the spindle fiber. So these fibers helps in the connection of the chromosomes. So these are lying at the opposite poles, isn't it? So this is all about the metaphase, okay. And in this phase, as the cell lies in the equatorial line, okay, that is in the that is all the chromosomes, they are aligning in an equal equator. So this plate, this is also referred to as metaphase plate. So this phase is also referred to as metaphase plate. So what are the various key features? You can write it down, that spindle fibers attached to the kinetochores of chromosome, and the chromosomes are moved to spindle equator and get aligned along the metaphase plate through spindle fibers to both the pole. So as you can see that spindle fibers are attached to the kinetochore. So see, this is the metaphase stage, right? So here you can see these are the spindle fibers, okay? So they are attached to the kinetochores, okay? And they are moved towards the opposite poles. So and here all the chromosomes, they are lying in the equator, isn't it? So this is also known as metaphase plate. So next let us learn about the anaphase. Okay, what happens in the anaphase? It is very easy that after the metaphase, all the chromosome, that is the centromere, it will split. The centromere splits and the chromosome moves towards the opposite pole, isn't it? So as you can see here, the centromere, that is the splitting of centromere took place and the chromosome moves towards the opposite pole, right? Yes. So see here what happens. On the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously and the two daughter chromatids, okay, two daughter chromatids, now referred to as chromosome. This is very important. Now the chromatid is no longer the chromatid, it is referred to as chromosome in the anaphase stage. And the father-daughter nuclei begin their migration towards the opposite poles. So as we have seen that they move towards the opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome is towards the pole and hence leading edge, with the arms of chromosome trailing behind. So it is nothing but the chromosome moves towards the opposite pole, right? So here what happens? The centromere splits, isn't it? And the chromosome, it moves towards the opposite pole. These are known as chromosome now. So this is now chromosome. No longer it is centromere, okay? So anaphase contains 96% of chromosome, all right? So yes, now let us learn the telophase. So what happens? Centromere splits, as we have already seen, the centromere splits in the anaphase stage and the chromatid move towards the opposite poles. So now let us learn about the telophase. So in the telophase, what happens? In the telophase, the chromosomes have reached their respective poles and decondense and lose their individuality, and the individual chromosome can no longer be seen, and chromatin material tends to collect in mass in the two poles, okay? So during this process, what happens during this process? So during the telophase stage, 1 minute. So during the telophase stage, so we can see like this structure. So now the chromosome start to reappear again, okay? So see here, you will see the chromosome, isn't it? So see, right? So the nuclear membrane also, it reappears. So the following events which are taking place in the telophase stage, you can see that the chromosome clusters at opposite spindle poles and their identity is lost as discrete elements, okay? And the nuclear envelope, it begins to form, and the nucleolus, Golgi complex and endoplasmic reticulum, we can see them in the telophase stage. So here the two daughter cells are being formed. How many daughter cells you can see here? You can see here two daughter cells, okay? During the telophase stage, two daughter cells are being formed. Yes. Now, what is this cytokinesis? You know that cytokinesis is a process after the telophase. After the telophase, the cell will move to the interphase again, right? So for that, the cell need to divide. So how the cell divides? The cell splits. That is the cytoplasm splits. Cytokinesis is nothing but the division of cytoplasm. So karyokinesis is the division of nucleus. You can write it down. Karyokinesis is nothing but the division of what? It is the division of nucleus, okay? Division of nucleus. And cytokinesis, it is the division of what? It is the division of cytoplasm. It is the division of cytoplasm of the cell. Now, how the cell divides? So, if this is the telophase stage, so see, this is the telophase stage. Here the cell divides. The cell need to divide, right? So, in case of animal cell, you will see like this, okay? That means the cell, it tries to pull them inside, okay? So, and we will see a burrow-like structure, okay? So, in case of animal cell, it is achieved by the appearance of a, furrow in the plasma membrane, okay? The furrow gradually deepens and ultimately joins the center, dividing the cell cytoplasm into two. So here a furrow-like structure we will see, okay? A furrow-like structure will be formed, and farther from this side also the cell will try to bring it closer, okay? So farther the cytoplasm, slowly the cytoplasm, it will be divided. So the cytoplasm divides and the cell becomes what? The cell that is splits. The two cell will be formed. Here the two cell will be formed now by the process of division of cytoplasm. So this is how the cytokinesis takes place in case of animals. Now talking about the animals, now how the cytokinesis takes place in case of plants? The plant cell also divides, isn't it? So in case of plants, what happens? In case of plants, they have a cell plate. They have a cell wall, sorry. So in case of plants, they have what? They have the cell wall, right? So they have the cell wall. So let's say this is the cell wall. So this is the cell wall. So the cell division takes place by formation of a cell plate, okay? Cell division takes place by formation of the cell plate. So in case of plant, the cell division takes place by formation of the cell plate. So this cell plate is further known as what? This cell plate is also known as middle lamella. The cell plate is also known as middle lamella. So that is how the cell will now separate and it will form as two individual cell. Okay, so the cell plate is responsible for separating the two cells. So see, the cell, in case of plant cells, the wall formation starts in the center of the cell and the cell grows outwards to meet the existing lateral walls. The formation of new cell begins with the formation of simple precursor called the cell plate, and that represents the middle lamella between the walls of the two adjacent cells. So see, this is how the cell divides in case of plant cell. That is the cytokinesis takes place. That is the division of what? Division of cytoplasm, which leads to the formation of what? Which leads to the formation of two new daughter cells. So yes. Now, the last topic, let us learn the significance of mitosis. What is the significance of mitosis? Why this mitosis is very, very much necessary? So, because it is responsible for the production of the diploid daughter cells. So, what is its function? It results in the production of diploid daughter cells, okay, with identical genetic complement. So, see, this is end cell, this is end cell. So after the fusion, what it will do? It will result in the formation of diploids. So the gametic cells are haploid, right? The gametic cells are haploid. So after the fusion of gamete, it leads to the formation of diploid daughter cells. And the growth of multicellular organism is due to mitosis. That is, it leads to cell division. Because of cell division, what happens? The organism will be growing, right? So it leads to the process of cell division. So in that process, the growth also takes place, right? And the cell growth results in distributing the ratio between nucleus and the cytoplasm. Next, it is playing a very significant contribution in the cell repair. So it is also responsible for the repairing of the cell. So if in our body any of the cell gets damaged, okay, now how the cell will divide? This damaged cells or the broken cells, it needs to further undergo cell cycle, cell division. So it undergoes this mitosis process and it helps in the process of cell repair. And the cells of upper layer of the epidermis, that is our skin cells, okay, cells of the lining of gut and the blood cells are being constantly replaced with the help of this mitosis. And the mitotic division is the meristematic tissue, okay, meristematic tissue, where we will see in case of plant, okay. So mitotic division in case of the meristematic tissue, that is the apical and the lateral cambium, results in the continuous growth of plants throughout their lives. So as you know that in case of plants, we have learned that the meristematic tissues are the active tissues, isn't it? So the meristematic tissues, we have learned that these are the active tissues, and there are three types of meristem, like apical meristem, lateral meristem, and intercalary meristem, isn't it? So what happens, how these meristematic tissues are active continuously? It is because of the process of mitosis, okay? So it is because the cell divides continuously. So it is very much significant, okay? So that is the reason it plays an important role in case of the plants as well as in case of animals. So this is all about it. In today's class, we have learned about the process, how the cell divides, what is the process, what are the various phases of cell cycle, and we have also seen the various stages of the mitosis and the significance of mitosis, right? So this is all about it. I hope you have understood what is being taught in today's session. And if you like this video, do like it and also visit to our website. You will be getting all the PDF notes of these topics. So in my next class, we will be learning about meiosis, which is another interesting topic. So be connected with Vista's learning. I will meet you in my next class. Till then, thank you and bye-bye.